Welcome to this tutorial on chemistry and reactions of carbohydrates, brought to you by Medical Biochemistry Power Capsule series. This video covers the following CBME curriculum topic: discuss and differentiate monosaccharides, disaccharides and polysaccharides, giving examples of main carbohydrates as energy, fuel, structural element, and storage in the human body. You may also go through tutorial 3A, which is available in this series. Carbohydrates undergo a variety of chemical reactions, each of which plays a crucial role in their metabolism, synthesis, and biological functions. These reactions contribute to the diversity of carbohydrate structures and their roles in living organisms. In the subsequent section we discuss some important chemical reactions of carbohydrates and their significance, such as glycosidic bond formation, oxidation-reduction reactions, hydrolysis, dehydration reactions, Maillard reaction, fermentation. And here's a funny mnemonic to help you remember the terms in this list of different types of reactions. These important chemical reactions of carbohydrates are central to their metabolism, synthesis, and biological functions. These reactions play key roles in energy production, structural elements formation, etc., highlighting the diverse roles of carbohydrates in living organisms and their significance in various fields, including nutrition, biochemistry, and food science. Glycosidic bond formation involves the condensation reaction between two hydroxyl groups, one from each of two monosaccharide molecules, resulting in the formation of a glycosidic linkage and the release of a water molecule. This reaction is central to the formation of disaccharides, oligosaccharides, and polysaccharides. Glycosidic bond formation is significant as it enables the formation of complex carbohydrates with diverse structures and functions. Formation of glycosidic bond has been illustrated below, in which two molecules of glucose has joined together to form one molecule of the disaccharide maltose. Carbohydrates undergo oxidation and reduction reactions, primarily involving the conversion of aldehyde or ketone functional groups to carboxylic acids or alcohols, respectively. In the box shown alongside, some examples of sugar acids and alcohols have been shown. You may want to remember the names of these compounds. Glucuronic acid, sorbitol, dulcital and mannitol have significant applied importance. Hydrolysis is the reverse of glycosidic bond formation, involving the cleavage of a glycosidic linkage by the addition of a water molecule. This reaction is catalyzed by enzymes known as glycoside hydrolysis or glycosidases. Hydrolysis of carbohydrates breaks down complex polysaccharides into simpler forms, such as disaccharides and monosaccharides, facilitating their absorption and utilization by cells. Digestion of starch by amylase enzymes in the digestive system involves hydrolysis of glycosidic bonds to produce maltose and glucose. Monosaccharides, when treated with concentrated sulfuric acid, undergo dehydration with the removal of 3 molecules of water. Hexases yield hydroxymethylfurfural and pentases yield furfural. The furfural derivative can condense with phenolic compounds to give colored products, which forms the basis of Molish test. It is a group-specific general test for carbohydrates. The Maillard reaction is a complex chemical reaction between reducing sugars and amino acids that occurs during cooking and food processing. This reaction leads to the formation of brown pigments, flavor compounds, and aroma compounds in cooked foods, contributing to their color, taste, and aroma. The Maillard reaction is significant in food chemistry and flavor development, influencing the sensory properties of a wide range of foods. The steps involved in a typical Maillard reaction has been depicted below. You may actually want to know how that beautiful brown crust is formed in your bread which you had for breakfast. Fermentation is a metabolic process that converts carbohydrates, such as glucose, into alcohol or organic acids under anaerobic conditions. This reaction is catalyzed by microorganisms, such as yeast and bacteria, and is used in various industrial processes, including the production of alcoholic beverages, bread and dairy products. Fermentation plays a significant role in carbohydrate metabolism, particularly in energy production in the absence of oxygen. Almost all sugars can yield fermentation products, some of which have been shown here. The next section covers few more interesting reactions that depict the versatility of carbohydrates and the basis of the diversity of carbohydrate-based compounds and complex biomolecules. When the hydroxyl group of the anomeric carbon of a monosaccharide is condensed with an alcohol or phenol group, it is called a glycoside. The non-carbohydrate group is called glycone. The first carbon atom in aldoses and second carbon atom in ketoses are known as anomeric carbon atoms. Digoxin is a cardiac glycoside, which is used to treat cardiac failure. Fluorhizin is a glycoside of glucose and fluortin and is used to precipitate renal damage in laboratory animals. Digitonin is a cardiac stimulant glycoside formed from galactose and digitogenin. Antibiotics, such as streptomycin and erythromycin, are also glycosides. Hydroxyl groups of sugars can be esterified to form phosphates, acetates, benzoates, propionates, etc. Sugar phosphates have tremendous biochemical significance. Metabolism of glucose within cells starts with phosphorylation. Glucose 6-phosphate and glucose 1-phosphate are important biochemical intermediate compounds of glucose metabolism pathway. Both these molecules have been illustrated below. Please try to understand the basis of their nomenclature. Amino sugars may form by substituting hydroxyl groups with amino groups. Generally, the amino group gets added to the second carbon atom of hexases. Glucosamine is the constituent of hyaluronic acid, heparin and blood group substances. Galactosamine is present in chondroitin of cartilage, bone and tendons. Manosamine is a constituent of glycoproteins. The amino group in the sugar may be further acetylated to produce unacetylated sugars such as N-acetylglucosamine, N-acetylgalactosamine, etc., which are important constituents of glycoproteins, glycosaminoglycans and cell membrane antigens. In mild alkaline solutions, carbohydrates containing a free aldehyde or keto group will tautomerize to form enediols. In enediols, two hydroxyl groups are attached to the double-bonded carbon atoms. In mildly alkaline conditions, glucose is converted into fructose and mannose. The interconversion of sugars through a common enediol form is called Lobry-de Bruijn-van Eckenstein transformation. In laboratory analytical work, various chemical reactions of carbohydrates can be exploited for qualitative and quantitative analysis. These reactions provide valuable information about the structure, composition, and properties of carbohydrates. In the next section, we shall talk about some chemical reactions commonly used in carbohydrate analysis. Molish test is used to detect the presence of all types of carbohydrates, including monosaccharides, disaccharides, and polysaccharides. In this test, the carbohydrate sample is treated with alpha-naphthol in sulfuric acid. The alpha-naphthol reacts with the carbohydrates to form a purple or violet ring at the interface between the sample and the acid. The formation of this colored ring indicates the presence of carbohydrates. Benedict's test is used to detect the presence of reducing sugars, such as glucose and fructose. In this test, the carbohydrate sample is heated with Benedict's reagent, which contains copper sulfate, sodium carbonate and sodium citrate. Reducing sugars react with the copper ions in the reagent, causing the formation of a brick-red precipitate of copper oxide. The intensity of the color change can be correlated with the concentration of reducing sugars present in the sample. Felling's test is similar to Benedict's test and is used to detect reducing sugars. It involves the use of two solutions, Felling's A and Felling B. When heated with a reducing sugar, Felling's solutions react to form a reddish-brown precipitate of copper oxide. The intensity of the precipitate can be used to estimate the concentration of reducing sugars in the sample. Seliwanov's test is used to differentiate between aldoses and ketoses. In this test, the carbohydrate sample is heated with Seliwanov's reagent. which contains resorcinol and hydrochloric acid. Ketoses react with the reagent to form cherry red or pink-colored solutions, while aldoses produce no change in color or a faint pink color. This color difference allows for the differentiation between aldoses and ketoses. PAS staining is a histological staining technique used to detect the presence of polysaccharides, glycogen, and glycoproteins in tissues after an elaborate reaction. A magenta-colored complex is formed which allows the visualization of carbohydrate-containing structures under a microscope. Hydrolysis reactions are used to break down complex carbohydrates, such as polysaccharides, into their constituent monosaccharides for analysis. Acid hydrolysis, enzymatic hydrolysis, or chemical hydrolysis can be employed to cleave glycosidic bonds and release monosaccharides, which can then be quantified using various analytical techniques, such as chromatography or spectrophotometry. Carbohydrate chemistry offers a fascinating realm of scientific exploration, rich with intriguing facts. In the next section, we shall be delving into some of these facts. This funny mnemonic given alongside will help you to remember the list provided here. Please pause and go through the mnemonic and correlate with this list. Carbohydrates are inherently chiral molecules, which means that they possess asymmetric carbon atoms. Due to this asymmetry, a monosaccharide may have different spatial configurations. Interestingly, natural carbohydrates predominantly exist in the D form rather than the L form. This prevalence of D sugars in biological systems is thought to be due to their evolutionary selection. Shown here are the two stereoisomers of the reference carbohydrate glyceroldihyde. The configuration of hydroxyl group on penultimate carbon atom determines D or L. In case of glucose, the fifth carbon is the determinant. Regarding different configurations and isomerism, when sugars differ from one another only in configuration with regard to a single carbon atom, other than the reference carbon atom, they are called epimers. For example, glucose and mannose are epimers which differ only with respect to carbon 2. Similarly, galactose and glucose differs only in carbon number 4. However, galactose and mannose are not epimers, but distereoisomers because they differ in more than one carbon atoms. In considering epimerism and distereoisomerism, the reference carbon is not considered. Reference carbon determines only D or L. Isomers have the same molecular formula, but differ in their spatial arrangement. The presence of asymmetrical carbon atom gives rise to optical activity. When a beam of plain polarized light is passed through a solution of carbohydrates, it will rotate the light either to right or to left, depending on the rotation. Molecules are called dextrorotatory when light rotates to right, or levorotatory when light rotates to left. Thus D-glucose is dextrorotatory, but D-fructose is levorotatory. Equimolar mixture of optical isomers has no net rotation, and these are known as racemic mixture. It's important to note that the D and L notation has no bearing with the optical activity. D and L refers to stereoisomerism. Asymmetrical carbon atoms give rise to stereoisomerism, and different stereoisomers may have different optical properties with regard to polarized light. The D-glucose isomer is incidentally dextrorotatory, while L-glucose is incidentally levorotatory. Glycosylation, the process of attaching sugar molecules to proteins or lipids, plays crucial roles in cellular functions. Interestingly, the specific glycosylation patterns of proteins can vary between individuals and even within different tissues of the same individual. These variations contribute to the complexity of the human proteome and have implications for health and disease. Glycosylation has implications in diabetes mellitus and its complications, particularly diabetic nephropathy. The spatial configuration with reference to the first carbon atom in aldoses determines the anomer variety. D-glucose has two anomers, alpha and beta varieties. Alpha D-glucose has specific rotation of plus 112 degrees, and beta D-glucose has plus 19 degrees with respect to polarized light. However, This rotation can change if a solution is kept for a period of time. Specific rotation of alpha-D-glucose can change from plus 112 degrees to plus 52.5 degrees, and that of beta-D-glucose can also change from plus 19 degrees to plus 52.5 degrees under certain conditions. This change in rotation with time is called mutarotation. The differences between alpha and beta anomeric forms are dependent on the first carbon atom only. Mutarotation should not be confused with dextro or levorotatory as discussed earlier, but both are related to optical properties. Glycosaminoglycans are complex carbohydrates found in extracellular matrices. They exhibit remarkable structural diversity. Their composition, sequence, and sulfation patterns can vary significantly, giving rise to a vast array of biological functions. Understanding GAG diversity is crucial for deciphering their roles in processes such as cell signaling, tissue development, and disease progression. GAGs have been discussed in detail in Tutorial 3A and Tutorial 9A. Carbohydrate-protein interactions play pivotal roles in various biological processes, yet they are often challenging to study due to their transient and dynamic nature. Recent advances in techniques such as glycan microarrays and molecular modeling have provided insights into the specificity and complexity of carbohydrate-protein interactions, shedding light on their importance in health and disease. Proteoglycans have been discussed in detail in Tutorial 3A and Tutorial 9A. Carbohydrates are not only important as nutrients, but also serve as essential components in various drugs and pharmaceuticals. For instance, antiviral drugs, such as oseltamivir and zanamivir, are derived from carbohydrate scaffolds and target viral neuraminases to inhibit viral replication. Glycobiology, the study of carbohydrates, has enlightened us about the immense importance of carbohydrates in the biological system and their crucial role in cancer, cell signaling, autoimmune diseases, etc. Researchers have developed carbohydrate mimetics for therapeutic purpose. These are molecules that mimic the structure and function of carbohydrates. These mimetics offer advantages, such as improved stability and bioavailability compared to natural carbohydrates. They hold promise for the development of novel drugs, targeting carbohydrate-binding proteins involved in diseases like cancer and inflammation. Rare sugars like D-lactose, L-galactose, and L-adose occur naturally in certain organisms and have potential applications in pharmaceuticals and food science. The field of glycobiology offers a deeper understanding of the language of carbohydrates within the body, opening avenues for innovative research and medical breakthroughs.
Thanks for watching this video. Our next tutorial in the Medical Biochemistry Power Capsule series is titled Carbohydrates, Digestion and Assimilation. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you have enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and share with your friends. And do keep watching our channel to remain ahead in the game.